Hello, everyone. Welcome to the Neuroanatomy lecture series. And today we will be talking about the neural tube development and its differentiation, as well as defects of neural tube closure. So the central nervous system initially appears at the end of the third week as a slipper-shaped plate of thickened ectoderm, and its lateral edges will soon elevate to form the neural folds, and the neural folds will approach each other in the midline to form the neural tube, which we can see in the last photo. Now, later on, the cephalic end of the neural tube will show three main dilations, and they're called the prosencephalon, the mesencephalon, and the rhombencephalon. And they can each be called either the forebrain, midbrain, or hindbrain. Now, by the end of the fifth week of development, the primary brain vesicles would have differentiated into 5 secondary vesicles, and each of these secondary vesicles will contribute to a different part of the brain. For example, in the very bottom, we have the rhombencephalon, which is a primary brain vesicle. It will differentiate and give rise to 2 of the secondary brain vesicles, such as the metencephalon, which will later on differentiate to give pons and cerebellum, and the myelencephalon, which will later differentiate to give the medulla oblongata. Now, the wall of a recently closed neural tube consists of neural epithelial cells, as we can see in the bottom right photo. Now, once the neural tube closes, these neural epithelial cells will give rise to a few layers. One of these layers is the mantle layer, which is this green layer in the bottom right. And this mantle layer will later give rise to the gray matter of the spinal cord, which is this yellow area in the left photo. And the outermost layer of this neural tube is the marginal layer, which is this blue layer, as we can see in the bottom right photo. And this will give rise to the white matter. So the mantle layer will give rise to the gray matter, and the marginal layer will give rise to the white matter. As a result of continuous addition of neuroblasts to the mantle layer, each side of the neural tube will show ventral and dorsal thickening, as we can see in the second photo. And the ventral thickenings will form the basal plates, which contains the ventral motor horn, and the dorsal thickenings, which form the alar plates, which contains the dorsal sensory horn. Now, between the ventral and dorsal horn, there's the lateral horn, which contains neurons of the sympathetic portion of the autonomic nervous system, and it's only present at levels T1 to T12 and between L2 to L3.
Okay, now we have the neural tube defects. The neural tube normally forms the brain, spinal cord, and the spinal column. Neural tube defect is the failure of the neuropores to fuse or abnormal fusion of the neuropores, which happens by the end of 4th week of development. Thus, there will be defects in the brain, spinal cord, and the bones covering them. There will also be a persistent connection between the spinal canal and the amniotic cavity. Neural tube defects are picked up in the ultrasound checkups by the doctor during pregnancy. It's also associated with low folic acid levels and diabetes during pregnancy. Neural tube defects are classified as open or closed, depending if the lesion is exposed to the environment or not. Examples of neural tube defects are SPINA bifida, which have 3 subtypes: SPINA bifida occulta, meningocele, myelomeningocele. Then we have anencephaly, and then the most rare and severe one, craniorachis. Craniorachis is a very rare neural tube defect where neither the anterior middle, or posterior port of the neuropore fuse. In this condition, there is no brain, spinal cord, skull, and spinal column. Spina bifida. It's the failure of the middle or caudal part of the neuropore to fuse. It is very important to note that the fusion starts in the middle of the neural tube of the neuropore and then continues anteriorly and caudally. Spina bifida, as we said, has three main subtypes. Spina bifida occulta, which is the closed and the mildest subtype of spina bifida. Meningocele, which is an example of open subtype of spina bifida, and myelomeningocele, which is the most severe and open subtype of spina bifida. Spina bifida occulta. It's the failure of closure of the caudal part of the neuropore with no herniation. So there is no herniation of meninges or the spinal cord. Spina bifida occulta is usually seen at the lower vertebral levels with an intact dura mater. It is also the mildest type of spina bifida, thus it is sometimes called the hidden spina bifida. It leaves a small gap in the spine, but no opening or pouch in the back. The spinal cord and nerves are usually normal. Hence, it doesn't cause any disability. The clinical presentation can be it can be really mild, just a skin dimple and or a tuft of hair at the level of bony defect. As we said, there is a skin dimple, thus it is covered. Hence, it's a closed example of spina bifida. Next, we have spina bifida with meningocele. It's the simplest and mildest form of open spina bifida. It is characterized by herniation or cystic dilation of meninges carrying cerebrospinal fluid through the bony defect, but no neural tissue is involved. Then we have spina bifida with meningomyelocele, which is the most severe form of spina bifida and an example of open spina bifida, where a sac containing the meninges along with cerebrospinal fluid and neural tissue, so part of the spinal cord, also herniates through the bony defect. This happens as the bones of the spinal cord don't form completely, thus causing part of the spinal cord along with its meningeal covering to bulge out of the opening, and it's not covered by skin. The person can have partial or complete paralysis below that level of the spinal column where the defect has happened. Finally, we have anencephaly, which is failure of the anterior neuropore to close, causing a serious birth defect where the infant is born without the skull, part of the brain, or the whole brain, the brain as a whole. It is lethal within the first year of life.